just a little background on Matt. He is a Washingtonian, that is the West Washington, and he got his degree, his undergraduate degree at the Western Washington University, and it was a double major both in biochemistry and in mathematics. And then he went to Cambridge, where he was in the golden years of the Guarenti lab at MIT, and he really was instrumental, I think, in along with Brian Kennedy, in shifting that lab to aging research, and did some of the seminal work on yeast aging, the sirtuin story, and a number of other things. He went back to Washington, University of Washington, and he did a postdoc there in genetics. And in 2006, he assumed a assistant professorship in the Department of Pathology. Recently, it's noteworthy that he got the American Federation of Aging Research Award for Rising Star in gerontological and aging studies, and certainly his trajectory has followed that. Since 2006, he's now the director of the Healthy Aging and Longevity Institute at the University of Washington and the co-director of the Nathan Schock Center for Excellence in Research on the Biology of Aging. A few other points about him is that he's been one of the leaders in yeast aging and uncovering the pathways that modulate longevity in yeast, and carried that on through these incredible genetic screens in yeast, which are still ongoing, to C. elegans largely, and the long-term effort, of course, is to move this forward into mice. And it's noteworthy that he was one of the first people to identify the TOR signaling pathway, this in yeast, as being a major pathway that modulates lifespan. And as we all know, that carries certainly on to mammals. I think it's also noteworthy to say that he has a large group working in his lab. I understand there are 35 or so undergraduates who are still in the lab, probably at this moment, tracking down other genetic anomalies that might give rise to extended lifespan. And also he's a distinguished visiting professor at Guangdong University in southern China, where undoubtedly he probably has another team working on these projects. So without further descriptions, I'm going to turn it over to Matt and look forward to his presentation. Just one reminder, because we usually have people at outside sites listening in, I'll keep reminding you when you have questions, and don't hesitate to interrupt and ask questions. That's the nature of this seminar. We like to have it as interactive as possible, to press the microphone buttons for those of you who have access to that. Okay, Matt, thank you.
All right, thanks, Jim. Thank you for the invitation. It's a pleasure to be here. So today I'm going to talk about sort of a turn that my lab has taken. 2 years ago, I don't think anybody in the lab was really thinking about mitochondrial biology, and it seems like over that time, several projects have sort of converged on the role that many of these aging pathways, how they influence mitochondria, and the role that mitochondria play in signaling back to the nucleus and to the cytoplasm. And so the stories I'm going to tell you about today all have sort of the common theme that they involve mitochondria and also the influence of mRNA translation on aging. But I'd like to start my presentations by showing this slide. Some of you have, I'm sure, seen me show this slide before. And I like it because it sort of illustrates something that we all recognize, which is that different people seem to age differently. I also like it because it's kind of fun to think about Keith Richards. And so for those of you who don't know who this is, so I'm sure the graduate students are like, who is this person? This is Tina Turner. She's an actress and singer. And this, of course, is Keith Richards from the Rolling Stones. And for the younger folks in the crowd, you may recognize him from the Pirates of the Caribbean movies. But he's sort of infamous for his lifestyle. And so I think this is a good illustration of not only how different people seem to age differently. So Tina actually is older than Keith. You wouldn't necessarily guess that by looking at them. But how it's not all about genetics, and it's a combination genetics and environmental components. And I think in Keith's case, that really illustrates the role that environmental choices can play on our apparent aging process. And in Tina's case, maybe there's some cosmetic concerns there as well. But this is something we all recognize, right? That different people, at least at the level of appearance, seem to age differently, okay? And so what I'm interested in is trying to understand what are these genetic and environmental factors that appear to modulate the aging process. And obviously, one of the reasons for doing that is that aging drives many important disease processes in developed countries. So most of the medically relevant diseases in developed countries are age-associated. Your risk of getting them increases as you get older. And by understanding this underlying aging process, the potential is there at least to delay the onset and progression of multiple age-related phenotypes at the same time. So is that even possible? Can aging be slowed? And I think sort of the best example of an intervention that seems to slow aging across many different organisms is dietary restriction. And I'm sure all of you are familiar with dietary restriction. This really just refers to the observation that reducing nutrient availability in the absence of malnutrition can extend lifespan across many different species. And I'm going to sort of use the terms dietary restriction, caloric restriction, calorie restriction interchangeably. So if you see DR or CR on some of my slides, I'm sort of referring to the same thing. Now, I'm happy to talk to people about the potential differences and important differences in those terms, but I'm going to use them interchangeably for my purposes today. The other point I want to make is a lot of the data that I show you will be in yeast. So this is survival curves in yeast, control and dietary restricted, looking at the percent alive as a function of age, which in this case is replicative aging in yeast, and I'll talk more about that in a minute. How many times can the mother cell divide? The point that I want to make about dietary restriction is that in yeast, this is accomplished by decreasing the glucose availability. So this is really glucose restriction. Okay, so normally the yeast media have 2% glucose. If we reduce that to 0.5%, and in my lab we usually use 0.05%, that leads to this sort of increase in replicative lifespan. Okay, and over the last few years, the field has sort of converged on a pathway that many of us think plays a role in the lifespan extension and health span extension from dietary restriction, and that, of course, is the TOR pathway. Now, I want to make it clear I'm not claiming that TOR is the only thing that dietary restriction is doing to affect aging, but we think it's an important part of how dietary restriction is affecting aging. And so there's lots of evidence supporting that. So I already mentioned that dietary restriction can extend lifespan in lots of different organisms. Inhibition of TOR signaling can extend lifespan in lots of different organisms. And TOR signaling is reduced by dietary restriction. So the arrows are all going the right direction. Dietary restriction turns down TOR. If we turn down TOR in normal nutrient conditions, that's enough to extend lifespan. And many labs in yeast and worms and flies have done genetic experiments that are at least consistent with the idea that inhibition of TOR signaling and dietary restriction act within the same genetic pathway to modulate aging. Another piece of evidence for this is that a substrate of TOR, S6 kinase, has also been shown to affect lifespan in yeast, worms, flies, and mice. So mutations in S6 kinase extend lifespan in all of those organisms. So it's not even just a single protein in this pathway, but we have two components of this pathway now that, at least in the major model organisms for aging research, have been implicated in lifespan regulation. And one of the real interests now is trying to figure out the mechanism. So how is TOR signaling affecting lifespan? And we don't know the answer to that. And it may be different in different organisms. But some ideas that have been put forth is we know that inhibition of TOR reduces global mRNA translation, and that's been associated with lifespan extension in many organisms. We also know that inhibition of TOR turns up this lysosomal degradation pathway called autophagy, and many people think that that's important for lifespan and health span extension. It's also known that inhibition of TOR reduces inflammation, and there's a large body of literature in the mammalian field that inflammation increases with age, and one of the things that dietary restriction does is decreases inflammation. Now, there are other possibilities as well, but I think these are at least three likely possibilities for how dietary restriction and TOR signaling are affecting aging. So, sort of big picture of what I've been trying to do over the last several years is to really try to ask, well, what else is out there that affects aging across many different species. The idea being that if a particular gene or environmental intervention affects aging in yeast and worms and mice, then there's at least a reasonable expectation that it may play a similar role in people. And so the approach we've taken has largely been on a genome-wide scale in yeast and C. elegans to ask, what are the genes that affect lifespan? And then we can look and see what's conserved. What are the conserved aging genes between these organisms and take those into mouse models. And this is a large collaborative effort that's been going on for many years now, including people at the University of Washington, Peter Rabinovich, Warren Ladegese, as well as Brian Kennedy at the Buck Institute. And so it's, as you can imagine, it's a long process. But we've been, I think, pretty successful, at least at this level, at the model or the invertebrate model organism level of identifying conserved aging genes. And so I'm going to tell you today about a couple of projects that have come out of the yeast part of this scheme, and then also a mouse project as well. So our approach in yeast, one of the things we've been doing, as Jim mentioned at the beginning, is trying to take an unbiased, make an unbiased analysis of the entire yeast genome for genes that affect lifespan. And we've done that by utilizing a tool that's available called the Yeast Deletion Collection. So this is a collection of about 4,500 single gene deletion mutants, all in a common genetic background. And our idea was, well, let's just measure lifespan for all of them and see if we can find things that increase lifespan. And so we've been doing that now for many years. And we've now got data for about 99% of the strains. So we now have at least some lifespan data for about 99% of the non-essential yeast genome. Yeah?
Is that on the 2%?
This is all under, well, most of it's under control conditions, yes. This is under 2% glucose conditions. And I will say, for a lot of them, it's a very small data set. So it's not as if we really know with a lot of precision what the lifespan is for all of these mutants, but we at least have some information about all of them, almost all of them, and we've got a lot of information about a subset of them. And just to clarify, for the yeast replicative lifespan, again, this involves how many daughter cells can a mother cell produce. So the daughter cells are removed from the mother cell, and typically that's about 25 for an average wild-type yeast cell. It can produce about 25 daughters. So that's replicative lifespan. So how is this actually done? Jim was correct. We have multiple sites that actually are doing this assay, some of them at this moment, I'm sure. So this involves manual removal of daughter cells from mother cells, and that means using a micromanipulator, which you might be able to see is attached to this stage on the microscope, and then there's a fiber optic needle that controls, and we take the daughter cells off of the mother cells every couple of hours. They'll divide. We count how many daughters does a mother cell produce. There are 3 teams doing this, my lab at the UW, Brian's lab at the Buck Institute, and then, as Jim mentioned, we have a lab in the south of China at Guangdong Medical College where people are performing this assay. I want to make the point that all of the strains are coded, so none of the people doing the experiment actually know the genotype or the environmental changes that they're analyzing lifespan for, and we've got pretty good throughput. So in my lab alone, we do about 1,200 mother cell lifespans per week. And so I've told you that the average cell divides about 25 times. You can do the math and think about how many micromanipulations that really is. It's a pretty big number. Okay, so that obviously has generated a lot of data. So this is a summary of where the genome-wide screen was a couple of months ago. I haven't updated the list since then, but it hasn't changed much. So these are the mutations that we're fairly confident extend lifespan. There are 105 of them on this list. That's over a large amount of mother cells examined, and a couple of points I want to make here. One is that you'll see a lot of things shaded in red. These are all things that map to the TOR signaling pathway. So we've pulled out a pathway here that affects lifespan in yeast, not just a couple of components. The other point I'll come back and talk more about these things in red in a minute. The other point I want to make, the genes that have asterisks next to them are conserved between yeast and C. elegans. So deletion extends lifespan in yeast, RNAi knockdown of the ortholog extends lifespan in C. elegans. And that to me is pretty exciting. So you can see some of those are in this pathway. Okay, but some of them aren't. And obviously those are very interesting to try to think about what the function of these proteins is and how they might be affecting aging across this large evolutionary distance. So the evolutionary distance between yeast and C. elegans is actually greater than the distance between C. elegans and people. So these are pretty divergent organisms. And it's maybe a little bit surprising that there's any conserved aspects of aging between these two organisms. Okay, so I'm going to focus it. Yeah, go ahead.
Do all of those genes have orthologs in the 2 species?
No. So many of them don't. I don't remember the exact percentage, but I would say a majority of the yeast genes do not, so it depends a little bit on how stringent you are in defining an ortholog. Many yeast genes do not have a clear ortholog in C. elegans. And in fact, this list has not, all of the ones with asterisks obviously do, but this list has not been whittled down to only those things that have C. elegans orthologs. Yeah.
Is it your feeling that aging in yeast is a cell autonomous effect, or is there?
That's a great question. So we know that it is. There are factors secreted from yeast cells in the chronological aging model that I'm not going to talk about today. That's how long can a yeast cell survive in a non-dividing state. So this is more like a model of replicative aging in a mitotic cell. There's a whole other model of yeast aging that involves post-mitotic aging, and we know that there are factors that are secreted from the cells that can affect lifespan in that model. We don't have any evidence for that in this replicative model. Yeah. Okay, so I want to focus a little bit more on this pathway because it's going to be important in the two stories that I tell you today. So this is largely loosely termed the dietary restriction pathway. I've told you already we think this is acting in part by reducing signaling through TOR. SCH9 is the yeast S6 kinase, so that's that other protein that's a substrate of TOR. And we think in yeast this is working at least largely through inhibition of mRNA translation, and specifically inhibition of the ribosomal large subunit, the 60S subunit. And one piece of evidence for that is that you'll see all these things that are RPLs here. These are ribosomal proteins of the large subunit. That's what RPL stands for. So from this unbiased genome-wide screen, we identified a whole bunch of components of the large subunit of the ribosome. What you also may notice is there aren't many, there's one, RPSs, components of the small subunit of the ribosome. So we think this is fairly specific for the large subunit of the ribosome. And I'm not going to go into detail on the mechanism. If you're interested, the reference is here. We think the reason that 60S deficiency extends lifespan in yeast is because that leads to paradoxically increased translation of this GCN4 transcription factor. So most things are translated less efficiently, as you might imagine if you don't have a lot of ribosomes around, but a few things, because of the structure of their mRNA, are actually translated more efficiently, and GCN4 is one of those things. And that's absolutely required for the lifespan extension in these cases, okay? The reason that this model is pretty complicated is because it's not completely required for lifespan extension from TOR or SCH9 or from dietary restrictions. So we've got these arrows over here about other things that must be going on, since we know it's not all about GCN4 in those cases. Okay, so that's the simple, complicated model for how we think this pathway is working in yeast. And I'll come back to different components of that as I go through my talk. So I'm going to try to tell you three interrelated stories. And the first one has to do with what we think is a signaling event initiated in the mitochondria that regulates cytoplasmic translation. And this is largely the work of Joe Delaney, who's a graduate student in my lab, and two undergrads that are working with him, Umema and Annie, who some of you met. She was out here interviewing for grad school a couple of weeks ago. And the rationale for this project was to try to understand how some of these longevity mutants are affecting lifespan. So I showed you a big list of genes. There are many approaches you could imagine taking to try to figure out how they're affecting lifespan. One approach we took, though, was to try to understand whether they were regulating stress in a way that was different from wild-type cells. So I'm sure you all know about the links between stress and aging. Many mutations that extend lifespan lead to different stress response profiles in various systems. And so we thought we would try to look at this in yeast. And the idea that Joe had was to take long-lived mutants and just assess their response to different types of stress to see if we could figure out, we could put things into groups based on their stress response profiles. So he did this for 43 of the published long-lived deletion mutants from that list I showed you, and he looked at 4 different kinds of stress. He looked at oxidative stress in the form of paraquat, proteotoxic stress, unfolding proteins by heat, DNA damage stress by MMS, and ER stress by tunicamycin. And the method that he used is something that we're sort of fortunate that we can do in yeast. We have this cool machine called a bioscreen that's an incubator, shaker, plate reader, all built into one. And so we can inoculate cultures of yeast into wells, into 200 wells. We can do 200 wells per run. put them in this machine, it'll incubate them at whatever temperature we tell it to, it'll shake them up, and it'll take OD readings every 15 minutes. And so we can generate these nice growth curves in an overnight experiment. So we can do that for 200 different replicates of whatever we want each night. And so the idea is to do that in different concentrations of the drug, or in the case of the heat following heat shock, and then look at the outgrowth kinetics and ask, do mutants have differential effects on growth relative to wild type under different conditions of stress? So this is just showing you a wild type and one of the ribosomal protein deletion mutants with different concentrations of tunicamycin. And as you can see, as the tunicamycin concentration increases, the growth rate is slowed. So the growth curve comes up in a delayed manner. And so, of course, we can actually calculate the maximal doubling time and all of that. But this is just a nice visual way to see what the effect of the drug is. The other thing you can see is that some mutants show less of a growth inhibition in response to the drug. So you can see these curves, as the drug is increasing, are much closer together in the ribosomal protein mutant compared to the wild type cells. And we can use that change in growth rate as sort of a measure of, it's not true stress resistance necessarily, but it's a measure of how the cell is responding to different types of stress. And so this is just the data set for tunicamycin. We've got comparable data sets for all of the other forms of stress I talked about. Yeah, oh, yeah, sorry. Yeah.
So so tunicamycin basically you are taking care for the ER stress, right?
Yes.
So you block the glycosylation pathway, right?
That's right. So tunicamycin is an inhibitor of glycosylation in the ER, and that's why it causes ER stress. Yeah.
So then how come how that message translate to your ribosomal, because this is a ribosomal mutant, right?
Yes.
So what
you want me to give away the punchline now? No, so I'll give you the simple idea for what we think is going on. We think that when you inhibit cytoplasmic translation, you become resistant to ER stress because there's a reduced flux of new proteins into the ER. It's a simple model. We don't know if it's right, but that's sort of what we're thinking is going on. Okay, so this is the data for tunicamycin across these long-lived mutants, and these are only the ones that showed a statistically significant difference in growth inhibition compared to wild type. These are, I think, 6 replicates of each mutant under each condition. And so what you can see is a lot of long-lived mutants are resistant, so they aren't inhibited as much by the treatment. Some are more sensitive. This is a control. This is a transcription factor called HAC1 that's involved in inducing the ER unfolded protein response. We put that in there because we expected it to be sensitive, and indeed, this infinity here means that the HAC1 mutants at this concentration didn't divide even once over the 36-hour experiment. So that's just in there to show that it's working as we expect. This is the RPL20B mutant down here. I'm not going to spend a lot of time on the specifics, although there's a lot of interesting data buried in here. One point I want to make, though, is for all of the stresses, we thought there might be a correlation between the magnitude of the lifespan extension and the degree of resistance to the stress. We didn't see that for any of them. There was no significant correlation, and in fact, in this case, you can see there's even a little bit of an apparent anti-correlation. So it turns out that at least among long-lived mutants, the degree to which they're long-lived does not correlate with their resistance to any one of these stresses by this assay.
And then how do you, thanks. How do you control for the drug getting in? I mean, is that something?
We didn't. Yeah. We didn't do it. So I mean, I think if we wanted to go back on a case-by-case basis and try to figure out the biology of why this particular mutant is sensitive or resistant, then we would obviously want to do that. But here we're just trying to do a survey and see if we could pick out patterns. Yeah. Okay, and that's what we did here. So this is, I mean, obviously it's a little bit tough to do cluster analysis when you've only got 4 conditions, but you can do it, right? So Joe went ahead and did some different types of clustering algorithms to try to ask whether or not these are each of the gene deletions here. We got things grouping together, and we got some interesting things grouping together, and a lot of the ribosomal protein mutants grouped together. You can't really see it here, but what I want to point your attention to is this subcluster or grouping here, where we have 4 ribosomal protein mutants. These are all, again, deletions in the large ribosomal subunit, and what caught our attention was this deletion here in AFG3, and the reason it caught our attention is, as far as we knew, it has absolutely nothing to do with the ribosome or cytoplasmic translation. This is a protease in the mitochondria. It's in the inner mitochondrial membrane, and it's highly conserved all the way from yeast through to humans. And it's involved in, as you might expect a protease in the mitochondria would be, mitochondrial protein quality control and proper functioning of the electron transport chain. So why then is this mitochondrial protease deletion based on stress resistance profiles, which I don't know if you can see it here, but this is resistance to tunicamycin, paraquat, and heat, and sensitivity to MMS. Why would that be clustering with these components of the cytoplasmic ribosome? It's sort of a mystery to us, and we wondered whether AFG3 might have a role in cytoplasmic translation. Even though all the data that's in the literature says this thing is exclusively in the mitochondria and it's a mitochondrial protease, we thought maybe it's somehow affecting cytoplasmic mRNA translation. And so these are two experiments to try to address that. The one over here on the right is just a cell cycle analysis. Things that have a defect in mRNA translation tend to accumulate in G1. It's very characteristic in yeast. You have a G1 delay in factors that have a cytoplasmic translation or protein synthesis defect. And indeed, we can see that relative to the wild type, we do have a higher percentage of cells that are in G1. More convincing, though, is this polysome analysis. So this is looking at actively translated mRNAs, looking at mRNAs bound to multiple ribosomes. This is a sucrose gradient. And so coming off the sucrose gradient, you get free 40S ribosomes, so small subunit, large subunit, the complete ribosome. And then these are actively translated messages out here. And they're more actively translated the farther they are out on the sucrose gradient fraction here. So this has 2 ribosomes bound to the same mRNA, 3, 4, 5, and so forth. So these are really highly translated messages out here. And what you can see is there are just less of them in the AFG3 mutant. Yeah?
Is there a change in the cell size when...
Yes, there is. The cell size becomes smaller. It's a good question. The ones that have translation defects. So again, that sort of goes hand in hand with this G1 arrest and decrease in translation. Yeah. Well, petites are a little bit different. I don't know. We've never quantified the size of a petite. So these are actually, I'm going to use the term respiratory deficient. So petite refers to a mitochondrial mutant that is slow growing, which I guess technically this would be. But it's pretty imprecise because many things that affect mitochondrial function are not petite. And nobody, as far as I know, really understands why that's the case. Okay, and petites can also be mutations in the mitochondrial genome versus nuclear encoded. So I'm going to say this thing is respiratory deficient. If you put it on a carbon source that requires mitochondrial respiration, it will not grow. Okay. Okay. The other point I want to make here is that the polysome profile does not look exactly like what we see for a large ribosomal protein mutant. Okay. So this is the RPL20B mutant, and it's a little bit hard to see. This is, again, from that paper that I referenced earlier. But in the ribosomal protein 20B mutant, there's less polysomes, so there's less highly translated mRNAs, but there's a huge decrease in the 60S subunit relative to the wild type, which is exactly what you would expect for a mutation that decreases 60S biogenesis. I just want to make the point, we don't see that in the AFG3 mutant. So there's no difference in 60S and 40S abundance, really, free 60S and 40S. It's really out here in the translated regions. And here's just the quantification of area under the curves. So that led us to ask the question then, is AFG3, so remember this starts by just looking at long-lived mutants. Is AFG3 affecting lifespan by the same mechanism as the ribosomal protein mutants, which I took you through this pathway that we think is at play, where you knock out something involved in 60S ribosome biogenesis, and that leads to differential translation of GCN4 and lifespan extension. And here's the data for the RPL20B mutant that requires GCN4 for lifespan extension. So the wild type in black, RPL20B here is long-lived. When you combine it with GCN4, it's no longer long-lived, okay? And the same thing is true in the AFG3 mutant. In fact, when we combine AFG3 with GCN4, we actually get this somewhat synthetic decline in lifespan. So just like the case for the ribosomal protein mutants, GCN4 is required for lifespan extension in this mutant lacking a mitochondrial protease. So it's a little bit perplexing, again, as to why mutation of this mitochondrial protease would somehow potentially be feeding into the same pathway. Now, what we don't know yet is whether GCN4 is activated in the AFG3 mutant. So we know it's required for the lifespan extension. We don't know that it's actually activated yet in the AFG3 mutant. So this is sort of our model right now, and I put the whole model up here because this is sort of my new sort of holistic model of yeast dietary restriction. I haven't talked about this side of things, so you can ignore it if you want to. It's a really interesting story, but I just don't have time to talk about it. But this side here is essentially what I just showed you before, where mRNA translation downstream of TOR regulates GCN4 to affect lifespan, and we think that the AFG3 mutant is somehow feeding into this part of the pathway, and our idea, which we don't have a lot of evidence for yet, is that there's actually a signal that's generated in response to certain kinds of mitochondrial defects that signals to the cytoplasm to regulate cytoplasmic translation. And we think that's independent of mitochondrial respiration. So this is not a question of not having enough ATP to make proteins. And the evidence for that comes from some suppressor analysis that we've done. So we've been able to identify spontaneous suppressors in the AFG3 mutant that restore growth rate but don't restore mitochondrial function. So that's how I know it's not an ATP deficit, or at least not an ATP deficit occurring because of a defect in mitochondrial function. So for those of you who haven't ever worked with yeast before, this is a tetrad dissection here. So these come from diploids that are heterozygous for the AFG3 mutation. And when you take a diploid and you sporulate it, you get out 4 haploid spores, okay? And 2 of those spores will inherit the AFG3 mutation, and 2 of them will be wild type. And you can see, based on growth rate, in each of these cases, there's 2 slow-growing and 2 fast-growing. So the slow-growing are the AFG3, the fast-growing are the wild type. So what we did was to plate AFG3 mutants, look for things that grew with a doubling time closer to wild type, cross those to a wild-type haploid to make a doubly heterozygous diploid for the AFG3 mutation, and then this suppressor. Okay, this is just a spontaneous suppressor. We don't know what it is. And then we can sporulate that. And if you do the math, what you can see is that this is segregating as if it's in a single gene. Okay, so we think we have a single gene suppressor, extragenic suppressor, that's suppressing the growth rate defect of the AFG3 mutant, and those would be these things in yellow here. And as I said, the cool thing is it doesn't allow the yeast to grow on a respiratory carbon source. So we haven't fixed the defect in respiration, but we've fixed the defect in translation, and you can see that based on the polysome profiles here. So we've separated the respiratory defect from the translation defect. We're doing the experiment now to ask whether these suppressors also block the lifespan extension. My guess is they will. The other thing we're doing is we've taken several of those spore clones that came up out of the tetrad dissection that we know carry the suppressor, and we've sent those for sequencing, and we've gotten some sequence back, and we've got about 6 candidates now that I think we'll be able to narrow it down and figure out what the suppressors are. So I'm optimistic that if this idea is right, that there really is a signal from the mitochondria to regulate mRNA translation, that these suppressors will help us flesh out that pathway. Okay, so now I'm going to move on and talk a little bit more about mitochondria, and it'll tie in somewhat with the story that I just told you. And this is mostly the work of Jen Schleit, a former grad student in the lab, and Simon Johnson, who's a joint grad student with myself and Peter Rabinovich. And this project, in my mind at least, came from the idea of, we all know about dietary restriction mimetics, right? Drugs that once we understand how dietary restriction works, we'll be able to make drugs that target those pathways and get all the benefits of dietary restriction without actually having to eat less. So obviously that would be, wonderful. A lot of people would think that's a great thing.
Excuse me, before you're moving on.
Sure.
But through this deletion screening,
Yeah.
you have this AFG3, but it's sort of like a mitochondrial chaperone. So do you have anything else that looks like that? That's the only one. Because it seems you can separate the translation and mitochondrial, and the mitochondrial is.
So we've gotten very interested in this mitochondrial chaperone pathway, and I'm going to talk about that now. But it's different. So we don't have, as far as I recall, nothing else in this list of confirmed long-lived things is clearly another mitochondrial chaperone, right? So it's not as if this is general for mitochondrial proteases. And in fact, there's a component that functions with AFG3. It's YTA12, I believe. They act in a complex. YTA12 does not show the lifespan extension of the AFG3 mutant. We don't know why. It's a mystery at this point. So it's not the case that generally perturbing mitochondrial protein quality control gives lifespan extension and translation defects. Having said that, we're, as I'll tell you in a minute, interested in this pathway because we think it interacts with dietary restriction or how cells respond to dietary restriction.
Likely that this long lifespan can tolerate mitochondrial defects.
Yes, I think that's right. I think that the reason those cells are long-lived is a non-mitochondrial phenomenon related to expression of GCN4. But you can still get lifespan extension in the absence of respiratory function in yeast cells. And we know that's true for dietary restriction. So it makes sense. That's pretty well established, at least in our lab, that you don't need functional respiratory chain to get lifespan extension from dietary restriction. So it makes sense in that regard. Yep.
So I guess what I was surprised about is your spontaneous suppressors.
Mm-hmm.
So when I was trying to find spontaneous suppressors in a gene that we were working with several years ago, that was a hard job. And I'm surprised.
What was the phenotype?
We're a growth on ethanol. So it's actually spontaneous suppressors of clock 1.
Okay.
And we found them, but they were very hard to find.
Huh.
And
You mean you didn't get many when you did the plating, or?
yeah, we had to plate a lot to get
Yeah, well, I mean, I think that depends on how many potential targets there are. It also depends on what the phenotype is. So these are pretty easy, right? Because we're just looking at things that grow better.
Doesn't that say you've got you got probably a lot of genes being suppressed there?
I don't think we know yet because so I've asked my graduate student who's doing that question, and he hasn't been careful about quantifying how many he's had to go through. I don't think we know yet. It's possible that there are multiple targets that can suppress this growth rate phenotype.
Was this just a random experiment, or was this?
No.
So these are all your hits, basically.
No, well, these aren't all of them, but this was a designed experiment to identify suppressors. We knew we had a growth rate defect, and we knew that we would get escapers from that growth rate defect, so we knew they could exist. So he went ahead and plated, 10,000 colonies and looked for colonies that came up faster.
Okay.
But we don't. You're right. It may be that there are multiple targets in the genome that we can hit to get suppressors. I think the sequencing will. We're not going to stop at one. I'll say that. Any other questions before I go on? Okay. All right. So dietary restriction mimetics, great thing. Okay, we can make a drug, give us the benefits of dietary restriction without having to eat less. And I really believe that this is possible. So don't get me wrong. I think that it's possible. These things may actually, rapamycin may be to some extent a dietary restriction mimetic. But I also think that there's a potential complication here, and that we don't really understand how individuals will respond to dietary restrictions. So we know that there is clearly, that genotype clearly plays an important role in the response to dietary restriction. But we don't have any understanding for why that's the case. Or I should say we don't have any understanding. We have a limited understanding for why that's the case at a molecular level. And that may be important. So if these dietary restrictions actually do become developed and widely available, they're probably not going to work the same for everybody, and they may actually be harmful for some people. And I bet that in this room, there are some people who dietary restriction would be harmful for. So we want to try to understand what's the interaction between genotype and dietary restriction, and what are the molecular mechanisms that drive that genotype by environment interaction. And our approach was to do this in yeast, in part because we can. And so we took a look at a series of mutants, and we just subjected them to dietary restriction and asked, what's the effect on lifespan? So this is 0.05% glucose. For some of these now, we've actually gone back and done a full dose response. But what's shown here is just a single level of dietary restriction. Each of these bars here is a different mutant. Most of these are single gene deletions. There are some double mutants and triple mutants in there as well. But that's not really important. I think the key point here is here's wild type. It's a fairly small lifespan extension from dietary restriction. We have many things that get a bigger effect on lifespan, and many things where dietary restriction is actually harmful, and some where we see no change from dietary restriction. And I should point out, this is the percent change in lifespan in response to dietary restriction. So this guy down here loses 90% of its lifespan when you put that cell on dietary restriction. This was strikingly similar to work that Jim and his colleagues had done in recombinant inbred lines of mice, where they subjected these lines of mice to dietary restriction. And even though the data is presented a little bit different, this is the absolute difference in lifespan from dietary restricted to control conditions. You can see the distribution is sort of eerily similar here, where there are some things that some of the strains benefited from dietary restriction, some of the strains were harmed by dietary restriction, and in some cases there was no obvious effect on survival. So I thought this was interesting and a very positive sort of outcome of our experiment. Yeah.
You mentioned that you were able to do some graded dietary restriction on some of these.
Right.
Has there been any pattern that's shown up with the negative responders or the positive responders?
So let me talk about that. I actually don't have the data in here. I should have put it in, but I'll try to talk about it in each of the cases that we go through. So one question that we were interested in, and we weren't sure whether this would be a complicating factor in our analysis, was, is it the case that the only thing that showed a big lifespan extension was long-lived, and the only thing that showed a big lifespan decrease was short-lived, or vice versa? Turns out, when you look at the data set as a whole, there's minimal correlation between, and it's not significant, between the percent change in lifespan from dietary restriction and the absolute lifespan under control conditions. So these are long-lived things out here. These are short-lived things down here. And you can see we get fairly large effects in both directions among both short-lived and long-lived things. Now, because this is a percent change, obviously it's easier to get a bigger percent increase in lifespan if the absolute lifespan, control lifespan, is shorter. But there's no correlation between absolute lifespan and the effect of dietary restriction. We've also gone and done growth rate analysis. So we also wanted to know, are slow-growing mutants more likely to have a positive effect from dietary restriction or a negative effect from dietary restriction? There's, again, zero correlation between maximum doubling time in glucose and the effect of dietary restriction in 2% glucose and the effect on lifespan of dietary restriction. So it doesn't seem as if health, this is sort of a measure of overall health of the cells, is a predictor of how those mutants are going to respond to dietary restriction. Okay, so one of the advantages we have, because we're working in yeast and most of these are single gene deletions, is that we know what all of these deletions are. So we can start to try to ask whether anything jumps out at us in terms of what are the functional defects among these good responders and bad responders. And so I'll start by just showing you a little bit of data on the negative end of the scale. And I'm not going to go into a lot of detail here, in part because we don't have a lot of detail, unfortunately. But one of the things that jumped right out at us, so there are several proteins that all act in the same complex. These are vacuolar membrane ATPase subunits. So there's an ATPase in the yeast vacuole, or the mammalian lysosome, that's required to maintain the acidic pH of that organelle. And so these are components of that ATPase. So in these mutants, we know that vacuolar or lysosomal pH homeostasis is perturbed. The pH of the vacuole increases in these mutants, so it's less acidic. And you can see they're short-lived to begin with. But when you put them on dietary restriction, their lifespan just drops dramatically. So it goes from a lifespan of about 8, which is short under control conditions, to barely even being able to divide 2 times under dietary restricted conditions. Part of the reason why we haven't really followed this up is because they're so short-lived to begin with that they're a little bit difficult to work with. So we're interested in trying to understand the mechanism, and I think it's intriguing because you could easily imagine how, if your lysosome is less able to handle cargo that gets delivered there under dietary restriction, that's going to be a problem. Because we know one of the things that dietary restriction does is it increases autophagy, which is going to mean more stuff gets sent to the lysosome. So biologically, this makes a lot of sense. But in terms of a molecular mechanism, we're sort of at the point where we're still trying to figure it out. Another interesting case that came out on the side of the negative responders are the superoxide dismutase enzymes in yeast. So there are 2 superoxide dismutases in yeast. Superoxide dismutase 1 is the cytoplasmic superoxide dismutase, and SOD2 is the mitochondrial, or primarily mitochondrial. And you can see the SOD1 mutant, even under control conditions, again, is very short-lived. So it has an average lifespan of about 4. You put it on dietary restriction, and it's pretty much dead. So again, we haven't really done much with SOD1 because it's so sick to begin with. SOD2, though, is interesting because you look out here and its average lifespan under control conditions is about 27, 28. So it's a little bit longer lived than wild type. You put it on dietary restriction and it drops by 90%. I think this might be our strongest negative effect. And so what are the superoxide dismutases? I'm sure everybody in this room, or most of you, know that these are enzymes that are involved in detoxification of superoxide radicals. They're, the mitochondrial superoxide dismutase in particular is important for detoxification of superoxide generated by the mitochondria and converting it to hydrogen peroxide. This is the lifespan data. So you can see again, the SOD2 mutant under control conditions is pretty much on top of wild type. There may be a slight increase in lifespan, but if you put it on 0.05% glucose, it drops dramatically. And so on this one, we have done the sort of dose response curve, and it really looks as if it's a gradual decrease. So at 0.5%, it's a little bit shorter than wild type. At 0.1%, it's even more short. At 0.05%, it's even shorter. And we can actually, we have a method where we can, that we sort of consider like 0% glucose, which is where we put the yeast on a completely non-fermentable carbon source like glycerol, and that causes the shortest lifespan of all in a SOD2 mutant. So it seems as if it's a gradual response to dietary restriction in this case. So how might this be working? So I've sort of mentioned already some of the things that we know dietary restriction does in a yeast cell. We know that it's going to inhibit TOR, and that's going to have multiple effects. Two of them are to turn down mRNA translation, as I've already mentioned, and to increase autophagy. And so those are two things that happen with dietary restriction. Another thing that happens in yeast is you actually get an increase in mitochondrial respiration. Okay? And this is because yeast on high glucose primarily use fermentative metabolism, and as the glucose becomes limiting, they shift over to a more mitochondrial respiratory-based metabolism. And so that may increase oxidative stress. I keep meaning to put a question mark here, but I haven't done it yet. That might increase oxidative stress, although we don't really know that's the case. So one obvious explanation for the SOD2 mutant is that it's this part of the pathway, right? That under control conditions, you don't really need as much mitochondrial superoxide dismutase because you're not making a lot of superoxide in the mitochondria, but when you calorie restrict and shift to respiratory metabolism, that becomes exceptionally important. And that seems to be the case. So we've done the experiments where we block autophagy or we turn down mRNA translation, and that doesn't affect this response to dietary restriction. So if you block autophagy, the SOD2 mutants are still shortened by dietary restriction. In contrast, if we block respiration, and we've done that a couple of different ways, that protects the cells against the effects of dietary restriction. So this is just showing you the average lifespan of control or dietary restricted cells in red. Different genotypes. So wild type, you get an increase from dietary restriction. SOD2 mutant, you get a decrease from dietary restriction. And these two are respiratory deficient double mutants. So HAP4 is a transcription factor that's required to turn on the respiratory enzymes under dietary restriction. And RIP1 is the risky iron sulfur protein in the electron transport chain. So both of those mutants are completely respiratory dead. And they allow now the SOD2 mutant to have its lifespan extended by dietary restriction. So not only do they protect against the toxicity, but they allow the normal lifespan extension. So that leads us to the idea that this is really, certainly is dependent on mitochondrial respiration. And we're still in the process of trying to really demonstrate that it's oxidative stress as opposed to some other byproduct of mitochondrial respiration. So this, I think, is interesting. I mean, whether this is relevant in mammals or in humans, I don't know. But you could certainly, again, imagine how there could be people out there with variants in their superoxide dismutase enzymes, and that dietary restriction might not affect them the same way as it would affect somebody who has a wild type version of the superoxide dismutase. So from a biological sense, it makes sense that variation in superoxide dismutase activity could influence the response to dietary restriction outside of yeast, yeah. And we do to some extent, even in the cases where we've looked. So the next set of mutants I'm going to talk about, they're on the other side where they have their lifespan increased by dietary restriction. But there seems to be this very sharp break point. So we go down to 0.5% glucose, and we don't really see any effect from dietary restriction. But when you go between 0.5 and 0.1, then you really start to see this steep change. So I think there will be differences in the shapes of the curve or the dietary response curve. Okay. So now we're looking at things that are in the positive response end of the spectrum. And one of the things we noticed right away is that many of the mutants that had the largest % increase from dietary restriction were nuclear encoded mitochondrial proteins. 5 of those I think are shown here, but there were several others. And so a couple of them we've focused in on are the 2 components of the mitochondrial prohibidin complex, PHB1 and PHB2. This is a mitochondrial porin, and this is a coenzyme Q biosynthetic enzyme, and I don't recall what FMP13 is. But the story I'm going to tell you has to do with the mitochondrial prohibidins, and we saw it as a positive sign that both components of this subunit were behaving the same way with respect to dietary restriction. So what are the prohibidins? There's a fair amount of literature on the prohibidins, and I'll apologize if anybody in here works on them, but my take is that despite the fair amount of literature on the prohibidins, we really don't know what they're doing, okay? So if anybody wants to speak up and tell me you know what they're doing, I would love to hear it. But I'm sort of still a little bit perplexed about what the prohibidins are actually doing. So what is clear is that they form a ring-like structure in the inner complex. It's composed of PHB1 and PHB2. This complex is, again, highly conserved yeast all the way up to people. The mutants have been shown to be short-lived in both yeast and C. elegans, and in terms of their function, the direct evidence for function is lacking. So there are phenotypes associated with the mutants that suggest that they involve regulation of mitochondrial proteases. In fact, these are thought to be negative regulators of that AFG3 protease I talked about earlier. I don't understand the connection yet, but I'm hoping one day that I will. They also seem to affect assembly of electron transport chain complexes. So in the mutants, there's apparently less efficient assembly of electron transport chain supercomplexes. But beyond that, it's not really clear what their molecular function is. This is just showing you the lifespan curves for dietary restriction. You can see you get a very nice, robust increase from dietary restriction in the prohibidin mutants. And so we can take the same sort of logical approach to understanding, well, why is it that the prohibidin mutants show this positive response to dietary restriction? Is it translation? Is it autophagy? Is it respiration and oxidative stress? And so we've done that using the same sort of approaches I told you about previously with the superoxide dismutase, and we're pretty confident it's not autophagy and it's not respiration. And we've got a lot of data on this, but here are just a couple of experiments that suggest that. So we can block respiration with this HAP4 mutation, and the prohibidin mutants still respond very nicely to dietary restriction, or we can block autophagy, and we've done this with several different autophagy components. These are absolutely required for macroautophagy, and you still get the lifespan extension from dietary restriction. Yeah.
So does the mutant, when you block that, does it affect the mutant's lifespan when you don't dietary restrict it?
No, it's a little bit hard to tell here, but they have pretty much the same lifespan in these double mutants under control conditions have pretty much the same lifespan as the single mutants under control conditions. For the prohibidins, so the prohibidins, as I said, are short-lived, and they cause these double. So the prohibidin mutation is epistatic to the ATG32 or HAP4 with respect to lifespan. So they go to where the prohibidin single is. Okay, so it's not autophagy or respiration in the case of the prohibidins, which left mRNA translation. Now, obviously, there's other things that happen in response to dietary restriction, but these are the ones that seem most likely. And we think that's the case in the prohibidin mutants. And the evidence for that is we can take mutations now that we think are in this DR translation pathway and ask, under control conditions, do those translation mutants phenocopy the effect of dietary restriction? Do they give you the same lifespan extension? And in both the case of the S6 kinase mutant and the RPL20B mutant, you can see that they give you at least most of the same lifespan extension that you get from dietary restriction in the prohibidin mutant, okay? So it's consistent with the idea that it's inhibition of mRNA translation. The twist in the story comes in that we think it's a different mechanism than inhibition of mRNA translation is extending lifespan in the wild-type cells. And one piece of evidence for that is the general translation inhibitor, cyclohexamide, does not extend lifespan in wild-type cells. In fact, it shortens lifespan in wild-type cells. So if it was the same mechanism, we would expect it to have the same effect in the prohibited mutants. In fact, what we see here is that cyclohexamide actually significantly extends the lifespan of both the PHB1 and PHB2, not to the same level as dietary restriction, but it does have a significant effect of both 50 nanograms per mil and 100 nanograms per mil, where it's shortening the lifespan of the wild-type cells. And we've done other experiments that support this idea, that if you remember, in the wild-type case, we thought it was because certain ways of turning down translation activate this GCN4 transcription factor. That's the mechanism for the wild type. Okay, so here's the model for what we think is happening in the case of the prohibited mutants as to why DR is having a different effect. And I think that this is sort of interesting because if this is right, it says that DR can extend lifespan by different mechanisms depending on the genotype. So in this case, we think that DR, acting at least partially through TOR or TOR signaling, decreases mRNA translation, and that's alleviating this mysterious defect in the prohibited mutant that's shortening lifespan. So what is that defect? Well, we were sort of lost until Simon did an experiment, which was really one of these fishing expeditions that are very hard to get funded, where we did a proteomics experiment looking at the mitochondrial proteome of wild-type or prohibited mutants under control or dietary restricted conditions. So basically grow up a bunch of yeast under these different conditions, purify mitochondria, and then take them to mass spec. And this was done with Mike McCoss, who's in the Department of Genome Sciences at the University of Washington. And so this is just relative sort of abundance of different peptides, each row being a different peptide in wild type, under control in DR, or either single prohibited mutant. And one thing you'll see is that there are some clear changes that occur in the prohibited mutants that don't occur in the wild type, and there are some changes that occur in all three. And this set of changes that occur in response to dietary restriction are what you would expect in yeast, an induction of respiratory and electron transport chain components, other components that are involved in mitochondrial metabolism. But what are these things? These look interesting. So these are peptides that are elevated in the prohibited mutants and suppressed by dietary restriction back to the wild type state. And so there's lots of interesting things in there, but the one that we've gotten most interested in is an elevation of these nuclear encoded mitochondrial chaperones. And the whole class of them seems to fall into this pattern where they're induced in the prohibited mutants under control conditions, and that's suppressed by dietary restriction. And among those, and you might not be able to see it, but among those are HSP60, HSP10, mitochondrial HSP70, several other mitochondrial chaperones. And these are really the sort of defining features of this response that people refer to as the mitochondrial unfolded protein response that has been described in C. elegans and in mammalian cells. If you look in the literature, it's kind of interesting. It's sort of referred to as yeast don't have this response. I would argue yeast have this response, and we found a couple of mutants that actually turn this response on. So one interpretation of this is that the prohibited mutants have mitochondrial proteotoxic stress under control conditions, and dietary restriction is alleviating that mitochondrial proteotoxic stress. There's other possibilities, but that's one potential mechanism for what's going on here. This is just a Western blot confirming the HSP60 levels. So I didn't know much about, in fact, I don't think I knew anything about this mitochondrial unfolded protein response a year ago. I know a little bit now, and so this is from a review by David Ron. So David Ron's lab has done most of the work in C. elegans on this mitochondrial unfolded protein response. And the way this works, it's really the biology of this is really cool. So the idea is that there are unfolded peptides or electron transport chain components that aren't in the complex where they're supposed to be. They're in the membrane. There are these proteases that cleave these misfolded proteins in the membrane. They get secreted through these channels to then turn on a signal that's sort of nebulous at this point that activates a transcription factor in the nucleus that turns on these mitochondrial heat shock proteins that then go to the mitochondria to help alleviate this proteotoxic stress. So in some ways, even though the players are different, the way this works is pretty similar to the ER unfolded protein response that we all sort of are more familiar with. And some of these factors are conserved between yeast and mammals, but really not many have been studied that well. So it's clear that the HSP60 and mitochondrial HSP70 play a role in both organisms, and that this HAF1 transporter plays a role, and the ClipX protease plays a role. But beyond that, it's not really clear what the details of this pathway are. It is interesting, though, because this this response has been implicated in aging by Andy Dillon's lab, where they showed that when you knock down components of the electron transport chain, so these mitochondrial knockdowns that Shane works on, that induces this mitochondrial unfolded protein response. And their model is that that's actually why these mitochondrial mutants are long-lived. That when you induce this unfolded protein response, that's part of the lifespan extension, and that the mito unfolded protein response is required for the lifespan extension. And the other cool thing about this paper is they showed that inducing this response in the neurons is enough to actually extend lifespan and send a signal to induce this response throughout the animals, or at least in the intestine. So there seems to be some signaling pathway here between the mitochondria to other cells in the organism. The other paper I want to mention has to do with the prohibitins, because I've been talking about the prohibitins so this last 15 minutes or so. Prohibitins have been implicated in aging in C. elegans 2 by the Taver-Narakis lab, where they showed, much like what we were seeing in the yeast system, that RNAi of prohibidin 2 shortens lifespan, but when you combine it with dietary restriction, you get this really big increase in lifespan. So it's not exactly the same, but it's similar to what I've been telling you with deletions of prohibitins, where we get this much bigger proportional effect from dietary restriction in yeast. So there might be some conservation at play here. And so we've looked and we've started to address this. And one thing we found is that knocking down the prohibitins in worms does induce this mitochondrial unfolded protein response in worms. So in this case, we used reporters made by the Ron lab of HSP6 and HSP60. These are transcriptional reporters where they drive GFP. And these are sort of the standard in the field for whether the mitochondrial unfolded protein response is on or off. So when these things are not induced, the worms don't glow. And in response to mitochondrial proteotoxic stress, they get turned on. And this is a control here. So this is something that the Ron lab had shown. This is CCO1, cytochrome C oxidase, in the mitochondrial electron transport chain. You knock it down, and you get induction of HSP6 and HSP60. And here's the PHB2. And you can see it's just as strong for HSP6, and it may be even a stronger inducer of HSP60 than the CCO1 RNAi. And importantly, this is specific, or at least fairly specific, for the mitochondrial unfolded protein response, because we don't see the cytoplasmic unfolded protein response come on. That's HSP16. So it seems like, just like what we'd observed in yeast, prohibidin deficiency in worms is inducing mitochondrial proteotoxic stress. Now, does it work the same way with translation? So I'd already told you that the Taver-Narakis lab had showed you get this additive increase or large increase in lifespan when you combine DR with prohibidin deficiency in worms. So we took the approach of using the S6 kinase mutant, and I've shown you data in yeast that this mutant in yeast gives this big lifespan extension in the prohibidin mutants in yeast. So we wondered whether the same thing would be true in worms. So this is showing you that deletion of RSKS1 suppresses, or at least reduces, the induction of the mitochondrial unfolded protein response by PHB2 RNAi. Interestingly, the RSKS1 mutant may even have a lower endogenous expression level of HSP60 in this case. We're still sort of working this out to see what the actual effect is here. But for lifespan, it's interesting. PHB2 mutants are short-lived in wild type. They go to actually increasing lifespan. So RSKS1 is long-lived a little bit relative to wild type. And then the PHB2 RNAi, in this case, actually increases lifespan. So just like what I was talking about in yeast, where you get a differential response to dietary restriction, here we're getting a differential response to PHB2 RNAi in this mutant with reduced translation. And the Kapahi lab and others have demonstrated pretty clearly that there is, in fact, a translation defect in this S6 kinase mutant. So there's parallels. Obviously, we don't know whether the mechanism is the same at this point, but there are interesting parallels here between prohibitins and their response to dietary restriction and TOR signaling in both yeast and C. elegans, as well as their role in modulating mitochondrial protein homeostasis in both organisms. So this is sort of our model for what's going on with the prohibitins. So I think I've hopefully convinced you that the defects in the prohibitins lead to mitochondrial proteotoxic stress, or at least induction of the mitochondrial proteotoxic stress response, and that inhibition of translation or dietary restriction can alleviate that stress response and extend lifespan. And again, our model here It's the simplest model I can think of, which is that this might really have to do with just the amount of protein that's being translated in the cytoplasm and imported into the mitochondria. And that it may be that by reducing cytoplasmic translation, you're importing less protein into the mitochondria, you're reducing the protein burden in the mitochondrial membrane and allowing the endogenous quality control machinery to keep up with the proteotoxic stress. The sort of interesting thing that I want to emphasize, though, is that this question of how the mitochondrial UPR is affecting lifespan. So I've shown you that in the prohibited mutants, we have induction of the mito UPR and we have short lifespan. There's no causality. We don't know that's why they have a short lifespan, but it might be. But in many cases, it's been shown that these mitochondrial knockdowns in worms extend lifespan and induce the mitochondrial UPR. So in that case, it causes extended lifespan, if you believe the Dillon lab, or at least it's associated with extended lifespan. And that may mean that the effect of this mitochondrial stress response is context dependent, the effect on aging. It depends on what else is happening in the system to whether that positively influences lifespan or negatively influences lifespan. And I'd really like to understand what that context is and how that's working. Okay, so I've hit you with a lot of information. Just to summarize what I've told you so far. So the first story had to do with the mitochondrial protease AFG3. I told you that it reduces cytoplasmic translation when it's mutated. So AFG3 promotes cytoplasmic mRNA translation. The deletion enhances stress resistance, 3 different kinds of stress, and it extends lifespan, and that's in a GCN4 dependent manner, just like the mutations in the 60S subunit. And I've also convinced you that genotype in yeast plays a very large role in the response to dietary restriction. We know about a few processes that are involved in that. The vacuolar ATPase is important, and superoxide dismutase is important for getting a positive response to dietary restriction. And that, at least in this one case of the prohibited mutants, you can get a much bigger proportional increase in lifespan from dietary restriction. Now, that may be just a suppression of the short lifespan. But in any case, the percent effect on lifespan is much bigger in the prohibited mutants. And we think this has something to do with the mitochondrial unfolded protein response. We see the mitochondrial unfolded protein response induced in both yeast and worms by prohibited knockdown. And that in both organisms, that phenotype and the short lifespan are suppressed by DR. And so we're working right now, obviously, to try to figure out, well, what's the exact relationship? Is it really just the import of proteins into the mitochondria? Where does the mitochondrial unfolded protein response come into this? And how does the mitochondrial unfolded protein response affect aging in both organisms? Okay, so any questions on that? Okay, so now I'm going to finish up with just a quick story on a mouse project that I'm pretty excited about. And this is the work of Valerie Wall, who's a graduate student in Karin Bornfeldt's lab, and Simon Johnson, who I've already told you is joint with myself and Peter Rabinovich. And the rationale for this came from the yeast studies that I just told you about, where we had several mitochondrial mutants that were responding quite positively to dietary restriction. And so that led to the idea, in my mind at least, which is, I admit, sort of a crazy idea, that maybe dietary restriction would be beneficial for mitochondrial diseases. Okay? And that's actually sort of the opposite of the paradigm, I think, that most clinicians take. So most clinicians think of a mitochondrial disease, one of the problems being a defect in ATP production. So the idea would be you really want to push more calories through the system than taking calories out of the system. Our data, from yeast at least, might suggest the opposite, that really a more beneficial approach would be dietary restriction. Now, there's lots of reasons why that may not work. One is that if you're in a weakened state already and you put dietary restriction on top of that, that could cause some sort of synthetic negative effect. In any case, this is an example here of a yeast mutant that corresponds to a human mitochondrial disease mutation. So this is the SHY1 gene in yeast. It's required for electron transport chain components. And this is similar to the human SIRF1, which is interesting for various reasons. This SIRF1 gene has been mutated in Leigh syndrome, and what we see is that, again, like some of the other mitochondrial mutants in yeast, we get this much bigger lifespan extension from dietary restriction in the SHY1 mutant than we get in the wild type cells. So there was a model of Leigh syndrome that the Palmater lab at the University of Washington had developed. It's in a different gene, but we thought, well, maybe we can use this model to test this idea. So, so that mutation is in the NDUFS4 gene, and that also causes Leigh syndrome. So Leigh syndrome is sort of a diagnosis that's made based on phenotype in people, and there are multiple mutations in different genes in different mitochondrial complexes that can cause Leigh syndrome. It usually affects infants and young children. It's got very short survival once the disease is diagnosed, usually less than a year. And right now, there's really no effective treatment at all. So these mitochondrial diseases are very severe, and there's really not much that can be done for the patients at this point. So this is the knockout mouse that I was telling you about that the Palmater lab had made. So this is in the NDUFS4 protein that's knocked out. And this is an NADH ubiquinone oxidoreductase. It's not really clear exactly how it's working. It's thought to be involved in stability and assembly of complex 1. So knockout of NDUFS4 does not lead to complete complex 1 deficiency. There's residual complex 1 activity, and the degree of complex 1 deficiency varies somewhat by tissue type, although I don't think that's really been carefully analyzed. So these animals are pretty sick. They're healthy for about the first month, and then they develop ataxia and neurodegenerative disease phenotypes. So the question is, well, the big question is, does dietary restriction benefit these animals? But we thought that probably wouldn't have much chance of working because they're so sick. So the approach we took was to use this drug, rapamycin, which is potentially a dietary restriction mimetic and is an inhibitor of mTOR. And I'm sure everybody here has heard a lot about rapamycin. So our approach was to do this by injection rather than using the food that's been developed by the Interventions Testing Program and by Randy Strong, and to ask whether or not injection of rapamycin every other day would have any effects on health and survival in these animals. And this is just showing you the blood levels over the 2-day period between injections. And we were starting treatment pretty early. This is about day 21, so right after weaning. And it looks pretty promising. So this is a small n, but we see a fairly robust increase in survival from rapamycin. And what's really neat about this, so the Palmater lab has a fair amount of historical data for over 100 animals in this model for survival. And so the longest that they'd ever seen an animal survive was right around the length that our animals survived, which means that more than half in this small pilot study of the rapamycin-treated animals lived longer than this historical data had ever seen survival. So I think this is a pretty strong and real result. And we also see improvements in function, although we haven't looked much. We're going back now and doing many more measures of health span. But at least by rotarod, it seems as if rapamycin is partially suppressing this defect. Now, by no means is this a cure, so the controls would be way up here. But it is improving function on at least this one measure of the rotarod test. And I rarely show movies, but I decided to put this in. So this is an example of the untreated and rapamycin-treated animals. These are old animals, and one of the reasons I wanted to show this is because unlike when Randy does this is not a resveratrol-treated mouse. I don't know if anybody gets that or not. This is actually an untreated control, and this is the rapamycin-treated animal. So you can see it's not a normal mouse. It's not a cure, but it does seem to be having a positive effect in this model.
When did you start the injections?
21 days. We're going back now and doing daily injections, and we're starting them at day 10, and we'll see how that goes. That may actually be pushing it too far. If we start to see animals die off, we'll go back. We'll do daily injections, but go back to the 21 days. Yeah, Arlen? Yes. Well, so these animals don't show the normal growth even in the untreated conditions. The rapamycin-treated animals are a little lighter. So we do see a decrease in body weight in the rapamycin-treated animals. That may be because we're starting them so early. But they maintain body weight longer, as you might expect if their health is being improved. So the curves cross back over probably somewhere around 50 days or so.
I don't know if these or not.
But have you ever considered the injecting the rapamycin to the pregnant mother?
I've had people suggest that before. I haven't really seriously considered. So I have a real dislike of going through the whole IACUC. I don't have a problem with the IACUC, but I have a dislike of going through the process. And and I don't really want to try and go through the process to do that. It's a good idea. So I mean, the question you're asking is an interesting one. If you could hit them, while they're still, in the mother, would that give you an even better effect? It might, but we haven't really tried to do that.
Matt, so all the subunits of the complex one are intact in this mutated?
So there's decreased amounts of complex 1 by blue native gel and supercomplexes.
Oh,
But the
supercomplex.
complex is there. So there's lower amounts, but there is complex there. I thought about putting the blue native gel in there, and I didn't do it. I should have. So there's a decrease in the amount of the functional complex that's there.
But when you do the CR,
We don't know. Oh, that's a good question. Yeah, so that's on the books, right? So this was a small pilot project. Again, as anybody who's worked with mice, I'm sure knows, but I haven't done that much with mice. It's been a real pain getting the breeder set up and everything. We're just now to the point where we're getting fairly large numbers of these animals, and we can actually go back and we want to do exactly that experiment. Look at the mitochondrial complexes by blue native gel, as a function of age, plus and minus rapamycin. So one possibility is rapamycin is somehow restoring complex 1 activity in these animals. My guess is that won't be what we see, but it would be interesting if that is what we see. Yeah.
So the mice that you're looking at can do better on the rolling rod and then the activity that you showed us because their nerves are better or their muscles are better.
Again, we don't know.
So there's no pathology.
That's in the next set. So this first set was just let them go to end of life. So then now we want to actually look at the tissues as they're getting older. Brain obviously is one place to look, but we also want to look in other tissues. One of the interesting things I think that we may uncover, we don't have enough animals yet, but so far, really the pathology that's been done in these animals has all been in the brain because that's what they're dying from. But I think we may start to uncover pathologies in other tissues as we're pushing the survival out. So that's one of the things we want to look at.
I was wondering if these mice become kind of oxytropic, so you could do a rapa plus glutamate, so it's TCA building components. If you gave them glutamate and rapa, they may even do better because it's kind of a
So why do you think that?
well, if you're feeding them a lot and they're trying to do respiration because they do have complex 1, though it's deficient, they may not be shuttling enough into the building components. And so you would have like a synthetic oxytropy.
So you think we've created a problem by giving them rapamycin? In that respect?
Maybe you would do better if you did
I see.
rapa plus glutamate.
Huh, that's interesting. That had never come across my radar. I'll have to think about that. Then I'll decide if it's a good idea after I think about it for a while. So I can tell you what we're thinking. So one obvious model is it's enhanced degradation of damaged mitochondria. So we know that, or at least it's in the literature, that rapamycin should increase mitophagy. And it's certainly, you could imagine how there may be some mitochondria that have more complex 1 than others in these animals, and that somehow mitophagy is selectively degrading the worse off mitochondria. So that's one possibility that we're considering. May also have something to do with the mRNA translation and proteotoxic stress that I've been telling you about in yeast and worms. We've looked at HSP60, and it turns out that HSP60 actually seems to be reduced in these animals, at least in brain, compared to controls. And we don't really understand why that's the case. As this has also come up, it could be that somehow rapamycin is restoring complex 1 activity. And then I actually think there's a decent chance that part of this has to do with inflammation. So we know that inflammation goes wild in these animals and that rapamycin has anti-inflammatory properties. And so one experiment that we're doing that I hope will help address this is we're going to, in parallel with the next rapamycin study, we're going to do tacrolimus. So tacrolimus hits the same protein as rapamycin, but it doesn't inhibit mTOR. It inhibits calcineurin, and it also has immunosuppressive and anti-inflammatory properties. So if we see the same effect from tacrolimus, then we can say this is really not due specifically to inhibition of mTOR, but it's probably more of an anti-inflammatory effect. I really don't know I don't know what to predict from that experiment, but I think it will be informative regardless of how it comes out, yeah.
So you've mentioned anti-inflammatory properties of rapamycin twice. Now, what anti-inflammatory properties are you talking about?
Well, this is not my area of expertise. What I'll say is it's widely, so it's an immunosuppressant. That is what its primary clinical use is to prevent organ transplant rejections. Most of those are in combination therapies with other immunosuppressants, so I don't know how, I think there's some question as to how strong of an immunosuppressant rapamycin is on its own, but it clearly can have immunosuppressive properties. It's how exactly it modulates immune function, I think, is a little bit unclear, and there's probably people in this room who can maybe address this question better than I can. So if there's anybody who wants to help me out, please do so. But it's widely talked about in the literature as an anti-inflammatory agent.
Well, I can help you out a little bit.
Okay.
In that. So we've treated mice with rapamycin from between 5 and 19 months. And this is used in Randy Strong's food formulation. And rapamycin actually can promote some measures of inflammation. And
Okay.
We're not the first people to have seen this. It's not immunosuppressive in anything that we can measure. It doesn't suppress any immune function we can find. It doesn't increase the numbers or function of any immune suppressive regulatory cells or cytokines that we can see. So when used as a single agent in otherwise normal mice, we don't see that it's anti-inflammatory. We don't see that it's immune suppressive.
Do you think that's dose dependent, or do you think that even if you were to ramp up the dose of rapamycin? I mean,
Well,
this is, I think, really important because
you're using doses that are way higher than what we would use. So the rapamycin levels in our mice are around let's say 10 nanograms per mil in the steady state, and yours were a lot higher. But
at least at some points in the dose response curve, yeah.
It went up and down.
Yeah.
The levels we use are in line with what you see in human patients.
Okay.
So,
Okay.
we could talk about
Yeah, no,
this.
I think this is really interesting because I think this is a really, really important point, right? Because one of the criticisms that people have to rapamycin as a potential, anti-aging or for age-related diseases is, oh, it's so bad because it's an immunosuppressant. And I think that those kinds of that kind of information is really important. And as we both have said, it's usually used in combination with other immunosuppressants. So yeah, so this is a model for what we think might be going on, but hopefully the tacrolimus experiment will help address that. Yeah.
Since your mutants still have residual complex activity, so the other obvious mechanism could be the increase the mitochondrial biogenesis, just increase everything, which can also rescue.
Yeah, that's a good point. It may be that there are just more mitochondria in response to rapamycin, and I guess there's some reason from the literature to think that's a possibility. I'm trying to think if we have any data on that yet, and I don't think we do. I guess we need to look. The one piece of data that might argue against that is that we see a decrease in HSP60. And so usually under conditions of enhanced mitochondrial biogenesis, you see an increase in HSP60, but we should look. That's a good idea.
The other part, just I noticed that under DR you list mitochondrial respiration independent of this mTOR pathway. But some people suggest that mTOR pathway can regulate mitochondrial biogenesis, but which obviously cannot induce oxidative stress.
Yeah.
But you have an idea which mediates respiration in your
yeast? Yeah, so in yeast it's pretty well worked out. So in fact, that's a good point. So inhibition of mTOR in yeast, and this has been, I think there's data in the literature and other model systems as well, certainly alters mitochondrial function, and there's some evidence that it increases mitochondrial biogenesis. And in yeast, it is true that the TOR1 mutant